lines. And the first, I will give an introduction to the PMN research strategy, and then I will give you some examples of glycans and nanoparticles for probing multivalent protein ligand interactions. These including the monolectins we have been targeting for DC sign and DC sign R, and then glycan quantum dots for probing MLGI affinity mode and binding thermodynamics. And also these nanoparticles for basically modifying for probing glycan go nanoparticles for probing MLGI and also block Ebola viral infections. And
Professor Zhou.
Yeah.
I'm just letting you know that you're on the presenter view right now. If you could possibly switch to presentation view, that would be really good.
Okay, let me see. Is that better?
Yeah, that's perfect. Thank you so much.
Okay, right. Yeah, and then the third part is going to be on gold nanoclusters for combating bacterial antibiotic resistance, and of course, the final is going to be on the acknowledgement. So, what is really a PMN strategy? So, what we do here is really to basically use nanoparticles as scaffold. We can play with surface and bioconjugation chemistries. In this case, so it can optimize and maximize the accessibility and function of these ligand and biomolecules. So in this case, we can make the nanoparticle to have polyvalency and also multifunctionality. Here I give you examples how we may be able to maximize the function and the accessibility of these biomolecule and ligand. Here is one of our examples of the ligand. We have the dicyl group, which can bind to this nanoparticle surface very strongly. We also introduce oligoethylene glycol linker that can provide water solubility, flexibility, and also preventing non-specific interactions. And also by choosing how the length of these EG linker, we can also improve this accessibility of these functional groups and the terminal end. So, and why multivalent binding is really important? If we consider from the thermodynamic point of view, if we have the receptor ligand binding and one-to-one interactions, then of course we can have these the binding and delta G, which is related to basically the association constant of the binding. Alternatively, we can also use the KD or the dissociation constant. And if we assume all these bindings are independent to each other, then the multivalent binding Gibbs free energy should basically equals n times the monovalent binding. And also the binding delta G is related to the minus RT log Ka, or if we use the KD, then the relationship is really in the log kind of relationship. If we convert it into the KD relationship, then the multivalent binding should be the monovalent binding's nth power. So that is fundamentally from the thermodynamic point of view. What does that mean? It's even for the monovalent binding, which is weak. For example, the KD is only 1 millimolar. If we have the bivalent binding, then that should give us 1 micromolar. If we have trivalent, 1 nanomolar, and also tetravalent, then that should improve their affinity to picomolar. So as you can see, basically by exploiting multivalency, it's a really very powerful tool to be able to enhance the binding affinities of weak individual bindings. But also in most cases, the delta GN is actually smaller the N times of the monovalent binding. So in that case, yeah, the overall KD for the N binding is going to be less than that. That is really reflected into the real situation that is in theory. Here is one of the examples that has been done in George Whiteside's group in Harvard. What they have target here, it's the antibiotics called vancomycin. It's a potent antibiotics against the gram-positive bacteria. It's specifically bind to the terminal end de-alanine by forming these 5 hydrogen bond. And the monovalent binding, it's about 1.6 micromolar for the KD. They then basically build a scaffold on the benzene ring, and each of them is linked to basically 3 of these vancomycin. And another, it's complementary. It's also built on the scaffold with the benzene rings. When they have the perfect spatial match, what they have found is for this trivalent ligand and the trivalent receptor binding, they found the KD here, it's 40 times 10 to minus 18. So effectively, it matches pretty well to what you may expect for this the monovalent Q. So the difference here, it's rather small. And for KD to be 40 antimolar, which is pretty much the strongest non-covalent interaction you have been able to achieve. Okay, so the third is we can integrate the unique functions and the properties of these nanoparticles. For example, here I give you an example of the quantum dots. Although all of these were made from the same material, cadmium selenide, and the size of the quantum dots increases from the 1.2 to 5.5 nanometers, their fluorescent emissions can be systematically tuned from blue to red under the same light excitations. Why this is the case? If you have a look into their basically absorption spectra, you can see all of these different colored quantum dots. They all absorb, has a broad absorption, which means if you use a 400 nanometer excitation light, you can excite all of them, while their emission is really controlled by their size, so they can be easily distinguished, and also basically their emission spectra is narrow and also symmetric and well. So in this case, as you can imagine, if you attack each individual quantum dots with a different target, then using one light source can allow you to simultaneously target several different targets. So that is really effective for multiplex sensing, imaging, and tracking studies. What we have done here is to use the fluorescence of the quantum dots and then coat it with a sugar, and then we can study their binding with the sugar binding proteins by labeling the protein with the acceptor dye. Then the fluorescence of the quantum dots can basically, if we excite the quantum dots, then it can transfer its excited energy non-radiatively through FRET, and leading to the dye emission from the protein. In this case, we can use them as a readout strategy to be able to determine basically how strong the binding are. This is a typical example. You can see basically the quantum dot fluorescence here and the dye emission here. If we enhance or increase the amount of the label protein added, then we can see the simultaneous quenching of the quantum dot fluorescence and the enhancement of the dye. If we use the ratio of the dye to the quantum dots, then against the concentrations, then we can fit the curve to derive how strong the binding are. And also quantum dots it's a solid nanoparticle. It has high basically contrast under electron microscopy. So in this case, if the binding cause a nanoparticle to aggregate or disperse, then we can use the TM to directly image to see basically how this binding, for example, in this case, it's binding to this sign that cause individual dots while binding to a different sugar binding protein that does cause the non-quantum dots to aggregate. So in this case, we can use them to derive what are the binding mode of each individual proteins as well. So these can be actually quite important for those applications. Okay, so when we have these PMN, because PMN can offer significantly enhanced affinity, then we can improve the sensitivity of the biosensing. So in this case, we can use them to achieve ultra-sensitive biosensing. We can also use them to develop multifunctional nanomedicine for serologic of cancer and also bacterial infections, etc. We can also use them as new biological tools to study the mechanisms of multivalent protein ligand interactions. And in this talk, I will be mainly focused on the multivalent protein ligand interactions, and also will basically introduce our work on basically antibacterial applications. Okay, so the first is the glycans and also the multivalent protein ligand interactions. Probably most of you all know, basically carbohydrates are really important for biology. And if you have a look into the red blood cell, you can see it's densely coated with these sugars. And these sugar coatings can be hundreds of nanometers thick. And in fact, our different blood group is really determined by this sugar content on the red blood cell. And of course, if you think about pathogen, virus, bacteria, if they want to infect cells, then they have to be able to navigate through these sugar coatings, and then basically has some kind of specific interaction in order to be able to enter the cells. And we also know for many of these protein sugar interactions, they are intrinsically weak, and monovalent level, they are non-bioactive. So in this case, if we can form multivalent binding, that can enhance the affinity and then allow the viruses and bacteria to enter the cells and of course to cause the infection. Here are two examples. The influenza virus, which use its surface trimeric hemagglutinin that is really bound to the citric acid on our cells in order to gain cell entry. And also E. coli bacteria, they also use their adhesion G and adhesion F to form multivalent bindings in order to attach to cells and of course to cause the infections as well. Because most of these infections are initiated through multivalent interactions, as you can imagine, if you want to block such kind of interactions, you probably do need to go to multivalent interactions as well. So here is an example. It's using the multivalent sugars to inhibit a bacterial toxin called the Shiga-like toxin. If you use only monovalent sugar, it's not very effective. Well, the IC50 is about 2 millimolar. If you can use the divalent, then the potency can be enhanced by about 40-fold. And if you have those basically pentadimeric, then the IC50 can be reduced to basically less than 1 nanomolar. So in this case, the potency had been improved by over 10 million-fold. And why this pentadimerics is so effective? If you have a look into their crystal structures, this is because these sugars, they have the perfect spatial match to 5 of these sugar binding domains. So they can simultaneously occupy all 5 in order to be able to completely block the bacterial toxin infections. In this case, the spatial and orientation match is absolutely essential for high potency. Yeah, unfortunately, the ones that we are interested is called DC sign and DC sign R. DC sign is one of those dendritic cell surface receptors, and they bind to virus and bacterial surface mannose and fructose-containing sugars to facilitate infection. And DC sign R is related to endothelial cell receptors. These 2 lectins are actually very similar. They both form tetrameric structures, contains 4 sugar binding domains initiated by the Lake region through the coil formation. And of course, they have those transmembrane part and then the cytoplasmal tail that give the internalization signals. They share 80% amino acid identity. They also share the same tetrameric architecture. However, their tetrameric structure is unknown, so that is really difficult to be able to target them really specifically. Although the tetrameric structure is unknown, monovalent CRD, the sugar binding domains are known. They bind to basically mannose in the same motif. If you see that DC sign R, it's mainly through the mannose. These are the 3,4 hydroxyl groups correlating to this calcium. That is a primary binding site. And you can see basically in DC sign, they are also the same kind of binding motif. But as the tetrameric lectins, they do have different glycan binding properties. DC sign does bind to these oligomannose, blood sugar groups, and also fructose-containing Lewis structures. But for DC sign R, it's mainly bind to the oligomannose. And more importantly, these 2 lectins are really involved in the HIV and HCV and also the SARS-CoV-2 transinfections. And yeah, in particular, DC sign, it's not more effective to transmit the HIV infection. And of course, HIV contains a GP120 trimer. Each of them are densely coated with mannose sugars. They only have a small number of the glycan spikes. And so in this case, the interspican distance is quite big. On the other hand, DC sign R, it's only DC sign R can transmit the wet slide virus. And in this case, each of those glycan proteins on the wet slide virus contain just 1 sugar. And they have a lot more number of those sugar spikes. As a result, their interspike distance are different. So in this case, clearly, as a tetrameric protein, they do seem to have different preference to the sugar density and also the intersugar distance. Unfortunately, their crystal structures are still unknown. So how their 4 sugar binding domains are arranged are still unknown. And this kind of information is really important if we really want to develop multivalent sugar ligand to be able to target them specifically. So in this case, we have first turned our attention into basically quantum dots. Why is quantum dots? As I mentioned briefly, quantum dot has strong fluorescence, so we can use coat the quantum dot with sugar, and then we can use its fluorescence to use a Fred readout we can also tuning the intersugar distance and then to probe how this intersugar distance will be able to change basically their spatial match and their binding affinity and specificity. We can also use different sugars, for example, the monomannose and also dimannose to find out how monovalent affinity really is affecting their multivalent bindings. And also importantly, because quantum dot is a solid ball, it cannot really deform, which means if one of the lectins that can bind simultaneously, then all of these sugar binding domain should be really facing in the right direction. On the other hand, if the other lectin is unable to bind strongly, which means their binding domains may be facing sideways, so it's not available for multivalent binding. So in this case, we can use this property to probe how these four sugar binding domains are spatially arranged. Also, if all of these protein bind simultaneously to one quantum dot, then if we have multi-protein bond, then that should give us individual nanoparticles. And on the other hand, if they cross-link, then we should be see them to assemble into much bigger structures. And these allow us to be able to directly visualize quantum dots to derive how their binding affinities. And also we can use the hydrodynamic size measurement to see, in this case, basically the assembly should be much bigger. So of course, there is an essential requirement is basically you have to really make your quantum dots sugar nanoparticles to be compact, because we need freight. Freight is very sensitive to the distance. We also need to have the dense sugar coating in order to enhance affinity here. So in this case, there has been a challenge because most of these high quantity quantum dots are prepared using the organometallic route. They are insoluble in aqueous media and bioincompatible. So in this case, they has to be kept exchanged with the hydrophilic ligand in order to be able to make them water soluble. Unfortunately, most of the literature methods, they do use a lot because their cap exchange efficiency is not good enough. They are using basically aqueous and organic biphase, and also use non-protonated ligand, deprotonated ligand, which has a relatively no binding affinity. So as a result, typically you need 10,000 to 100,000 of the ligand to quantum dots in order to be able to make the complete cap exchange, which is becomes really difficult to achieve with this precious ligand. What we have come to an idea is with our glycan ligand. Here we first use the sodium hydroxide to deprotonate the cell to make them silate. Silate bind much stronger to the quantum dots. We also perform the cap exchange in a homogeneous solution. So as a result, we can achieve significantly higher cap exchange efficiency. And under our conditions, we can reduce the ligand amount by almost 200-fold, which means we can use about 1 milligrams of the ligand to allow us to make about 1 nanomole of these quantum dots, which is really beneficial. We have also been able to make them each quantum dots to coat it with several hundred copies of the sugar. So that is really useful for strong binding. And also we found these quantum dots are relatively small. Their hydrodynamic size are still less than 10 nanometers. So they should act as good basically freight donors. In order to be able to use for freight readout, then we introduce a system mutation to the sugar binding domains just outside the sugar binding domain, so it's not going to affect the sugar binding properties. And then use a monoamide to label them with the dye, and then we confirm the labeling using basically the mass spectrometry. That is before and after labeling. You can see there's an increase of the molecular weight by about 850 Daltons. That it corresponds to the label dye, and the labeling efficiency here is about 80%. So when we have the quantum dots and also the label protein, then we can do the titration experiment. We have the fixed amount of the quantum dots and then titrated in different amount of the label protein. We can see basically the quantum dot fluorescence get quenched and the dye fluorescence is enhanced. That is for DC sign. For DC sign R, the quenching and enhancement is much smaller. And if we use a smaller and shorter linker, then the quenching and enhancement is significantly enhanced. If we use basically the ratio of the dye to the quantum dot, which is proportional to the amount of the protein that has bound to the quantum dots, and then versus the concentration plot, or we can see for DC sign that is short linker and long linker, they follow a typical binding curve. But for DC sign R, it's pretty much still on the baseline, which means DC sign R binding is very weak. And importantly, we found the ratio here for the short linker, we can reach 60-fold, and which is really unprecedented because initially these two lectins were thought to be the same. We have also been able to roughly estimate how strong the binding are. We found it's in the sub-micromolar range, so the KD roughly corresponds to half of the maximum binding there. Based on this result, we show basically DC sign clearly might be able to basically bind simultaneously or multivalently to each one individual quantum dot. So their binding domains should be facing upwards. But for this sign R, it does only show minimal binding, which means their binding domains probably split up and facing outwards. Because their monovalent binding are identical. So the difference must be related to the different orientations of their different binding domains. And also this work has been highlighted as a cover in the unwanted shimmy. This simply just show you have to have the four binding domains to work together in order to be able to bind strongly to the sugar-coated quantum dots. Although with the monomers, we have been able to differentiate DC sign from DC sign R, but DC sign R, it's really, we cannot differentiate with the monovalent CRD. So exactly how the binding appears, we still don't know. So in this case, we turned our attention to basically the dimanose, and in that case, the dimanose, the monovalent affinities about 4 times stronger. And with the dimanose, we do the same stuff. And then we find now, basically with the longer linker, we can differentiate DC sign R with the monovalent CRD and also DC sign. For DC sign, we see basically, initially it's a linear increase and then reach saturation. For DC sign R, it's a really S-shaped. Initially, it goes very weakly and then increase faster and then reach saturation. This is a clear indication of the positive binding cooperativity. And using this, we can fit the curve and then find out really the Hill equation here, and it's greater than 1, which means it's really for DC sign R, it's positive cooperativity, which means it's most likely to be caused by the cross-linking kind of interaction. So based on all these, we thought DC sign use all of its 4 sugar binding domains to bind to 1 quantum dot, while DC sign R use 2 of them to bind to 2 separate quantum dots. and then cause a cross-linking. For these different binding mode, then we should be able to see the difference when we use measure the hydrodynamic size, for example. Initially, with the quantum dots, then we do see individual and also basically slightly clustered. Binding to this sign, we see all of these clusters has completely disappeared from the TM images, and also the nanoparticle size, hydrodynamic size correspond well to basically quantum dots coated with layer proteins. But for this sign R, then clearly it's for much bigger assemblies and also confirmed from the TM images. These different binding, of course, it's really important to be able to figure out how strong the binding. Typically, these are through this titration experiment where you titrate in different amount of the protein and then to figure out basically the half binding by the Hill equation. However, these methods can have a problem. It's because although these methods can work well, if you have the weak binding, which means the KD is much greater than half of the concentration of the quantum dots times the number of the protein that can bind to basically each individual quantum dots, or equivalent to basically the saturation amount of the protein required to occupy half of the quantum dots binding capacity. If you have a stronger binding, then it's not going to be work, because all of these measures KD is going to be equals half of your quantum dot saturation binding capacity. To overcome this problem, we have used different methods. We use one-to-one ratios for the protein, and then simultaneously increase their concentration, and then adjust the instrument sensitivity to compensate the fluorescent intensities. Although this will basically change the absolute signal for both quantum dots and the dye. However, we're using the Freight ratio. It's this to this ratio which will not be affected. So using this method, we can use the ratio versus concentration to derive how strong the binding are. This method can work for both strong and weak bindings. Using this method, we found the binding here. It's quite strong. With the short linker, with the diamonds, we can achieve sub-nanomolar kind of affinity, which is a 1.5 million-fold stronger compared to monovalent binding. And also, even if you normalize to per sugar terms, then the enhancement is almost about 4,000-fold. We have also found basically for these diamonds bind to this sign, it's much stronger compared to basically this sign R. That can also basically explain why basically this sign is more effective in transmitting the HIV, because the HIV is densely coated with mannose sugars. Not only we can use them to basically measure the binding affinity, we can also use them to measure the binding thermodynamics. So in this case, we repeat the experiment under 3 different temperatures using the methods above, and then to find out how strong the binding are affected by the different temperature. If you can see for this sign and this sign R, both of their binding affinities become weaker when the temperature is increased. Then we can combine basically the Gibbs free energy, the relationship, combine these 2 equations, and then do the Vathoff analysis using log KD versus the inverse of the temperature. We should get a linear relationship. The slope here corresponds to the delta H divided by R, and the intercept should be the minus delta S divided by R. And that is the Vathoff plot. and that here is a result. We found both DCSI and the DCSI-R binding are enthalpy driven. So the delta H is about minus 100 kilojoules per mole. Include this is about 4 times the monovalent binding, suggesting all 4 CRDs are involved in binding. And also basically the delta S is also selective, which means this is enthalpy driven, but it's the bindings incur an entropy penalty, and the entropy penalty for DCSI-R is stronger, which is bigger, which means the binding is weaker. And also we found basically a small change to a domain that link to the binding domain, and the link can completely change its binding thermodynamics from the enthalpy to entropy driven as well. We also found actually these nanoparticles or the sugar-coated quantum dots can really potently block DCSI mediated viral infections. This is because if we have the sugar-coated quantum dots, then that will occupy the surface receptors on the cell. Then it will be able to prevent virus further attachment. And in our case, we found the IC50. It's about sub-nanomolar kind of level. But for DCSI-R, it's ineffective, which probably also consistent to its basically much weaker binding as well. Because quantum dots contains ketamine, so it's not really that biocompatible if we really want to use for biomedical applications. So here we have changed to the scaffold to basically a gold nanoparticle to make them more biocompatible. And also gold nanoparticle can have a much wider size and shape range, which can allow us to be able to further probe how shape and size affect the binding. And also it can have strong fluorescent quenching. We can use them to basically monitoring the binding affinity using the gold nanoparticle-based fluorescence quenching here. So in this case, we directly excite the dye on the protein, and the fluorescence is going to get quenched. And here, the solid line, it's a different concentration without the protein, and the broken line is with the same concentration of the gold particles. You can see basically without protein, then it goes linearly. But with the nanoparticle present, then yeah, it becomes sublinear and deviate more and more, which means it's an enhanced quenching here. So by using the quenching efficiency versus the concentration, we can also derive how strong the binding are. And also use the same strategy. We found bind to DCSI that give individual particles, and also bind to DCSI-R that's called extensive cross-linking of these nanoparticles. So these two proteins retained their simultaneous and cross-linking kind of binding mode. We have further compared their binding with their ability to inhibit viral infections. We found these particles are very effective to be able to block DCSI mediated infections. So in that case, if you see the inhibition curve decays very rapidly with fitting this equation where n equals 1. But for DCSI-R, and it's about 0.5, which means it's not really very effective. This is presumably on cell membrane surfaces, these cross-linkings become much more difficult to achieve. So the virus can easily basically still accessible. And also this work has been highlighted on the cover in JAX and well. Okay, so here is a comparison to how effective are glycan nanoparticles compared to the best of the literature. In our case, we found basically with the glycan quantum dots, a gold nanoparticle, our potency here, it's pretty much 10-fold better than the base literature reported previously. And also importantly, these can the entry inhibitors for these nanoparticles can basically prevent virus mutations and develop resistance. So this can be a really good advantage. Okay, so here is a summary to what we have learned from the galactic nanoparticles. We can discriminate subtle differences in these closely related multivariate. We can quantify binding affinities, thermodynamics, and kinetics. We can reveal their key structure information by combining freight TM and DLS analysis. We can also potently block virus infections. And also basically we revealed basically the binding mode here. It's really key in order to be able to design virus infection blockers. Okay, so the second story is going to be on the antibacterial nanoclusters. And according to the World Health Organization, bacterial antibiotic resistant, or BAR, is one of the biggest challenges to the global health. And in 2019, there are about 5 million deaths associated with antibiotic resistance. And also over the past 20 years, new antibiotics approved are significantly reduced, and bacteria become more and more resistant to basically frontline antibiotics. This make bacterial infection more and more difficult to treat. If the current trend continues, then by 2050, the annual global deaths of bacterial resistance will top up 10 million, which will overtake cancer as the most deaths and cost the world economy by over $100 trillion. Yeah, unfortunately, although antibiotics are very effective initially when they were introduced, bacterial resistance is really unavoidable. And this is due to the evolutionary adaptation. And they can actually appear very quickly. For example, penicillin, that is the first antibiotics introduced in 1941. And the next year, resistant bacteria has already been reported. For example, even for the latest ones in the same year, resistant microbials were also reported. And yes, most of these antibiotics, they typically use one of those antibiotics mechanisms. They can inhibit cell wall synthesis, inhibit protein synthesis, DNA synthesis, or inhibit folic acid synthesis, and inhibit RNA synthesis, etc. So most of these are simply offer one mode of the antibiotics mechanisms where bacteria can relatively easy to develop resistance. So what are these things, mechanisms? For example, you can, bacteria can develop and express enzymes to degrade antibiotics. For example, beta-lactamase against penicillin and related. They can also modify the target, make them much less effective for the antibiotics. It can also develop strategies to evade or bypass immunity. It can also efflux pumps to basically remove any antibiotics that has entered into the bacteria. And it can also form colonies in the form of biofilms that can exclude or reduce antibiotic diffusion. Bacteria can also increase resistance and also increasing multi-drug efflux pumps, and also activate hormone sensing as well. Of course, antibiotics nanomaterials has been a very active research area, and there are many research that has been done despite of this progress. Most of these research has been really focused on in vitro kind of properties, and only a few have demonstrated in vivo activity, and only a handful has really entered into clinical trials. Here is a few of the examples that have been in clinical trials. So the success rate for the antibacterial nanomaterials, it's really limited. This is related to challenges. How can you really precisely control the size and its formulation? Because most of these nanoparticles have different size, shape, etc. And also importantly, you have to have the suitable body clearance, and also they have to have non-toxicity, biocompatibility, and also good biosafety in order to be able to save for human use. So in our group, we have turned our attention to basically these small gold clusters called the gold 25 and SIL18 kind of type of this ligand. This is because these clusters have exceptionally high stability. And so in our case, we have selected to use a different type of the ligand. Instead of most other research has been simply just use one ligand. Here we introduce a pyridinium ligand that contains C12 chain, which means it's positive charge, so it can disrupt the membrane potential of the bacteria, because most bacteria is negative charge, and also it contains hydrophobic chain, which can disrupt the membranes. We also introduce a C5 chain, basically tritiline ligand, because this tritiline ligand is strongly hydrophilic, has high biocompatibility, and also high in vivo stability. It can also minimize non-specific binding. As a result of these small particles clusters, they should be easily cleared from the body, which means to avoid long-term accumulation, as it's also autonomy and formula precise. These are good for quality control, and we use two different ligands that can tune their antibacterial and biocompatibilities by simply varying these two ligand ratios. First, we basically screen to see how the feeding ratio of these pyridinium ligand affects their antibiotic properties. What we have found here is in this case, we are comparing their MIC, that is a minimum inhibition concentration. So the lower the MIC, the better the antibiotics. What we have found here is initially, of course, it's non-active, and about 45% to 50 that give a minimum, and then it becomes less effective. And also when we have 100%, then that appears to be effective again. So if here clearly we showed its antibacterial potency, it's not linear to the P12 ligand. We have then basically evaluate their biocompatibility by comparing their 5% lysis of the red blood cells. That is an indication of the blood compatibility. And then we use the SD5 to the MIC ratio to indicate the therapeutic window from the injection. What we have found here is the ratio here, it's the best when we have 45% of the P12 feeding. And this gives us the optimum kind of main product is each gold cluster contains about 10 C5 and 8 P12 ligand. So that is, in this case, we can have the optimum antibacterial potency and also good biocompatibility. So here we investigate what are the mechanisms. As a control, basically, it's simply just treated with PBS. We see these bacterial cells are intact. When we treat it with the antibiotics, And yeah, the shape does not really change. But when we treat it with the gold nanoclusters, you can clearly see these bacteria are aggregated, and also the membranes are really disrupted. And also, if we have the antibiotics combined with the gold nanocluster, the aggregations or the disruptions are even greater. And also, we can use the fluorescence of the gold cluster its own to monitoring basically how their mechanism as well. And also from the TM images, it's also show it's really disrupting its membranes. Because the disrupting of the membranes, then that should allow these antibiotics diffuse much more easily. So in this case, we have screened the combination against the multi-drug resistance bacteria, MRSA. In this case, if you only have the antibiotic on its own, we really need a high concentration, 256 microgram per ml, in order to prevent bacterial growth. When we have 8 microgram per ml of the gold cluster, then 2 microgram per ml of the antibiotics can completely prevent the bacterial growth. And also from the plate assays, we clearly show the combination can completely prevent the bacterial growth. And also, if we count the bacteria, then with the combination, we can reduce the bacterial count by 7 orders of magnitude. And indeed, we do find that the combination treatment can allow basically much faster recovery of these bacterial infection in the skin infection model. That's a control with the antibiotics on its own. And then with the combination, you can see basically under 12 days, then effectively all of these infections are almost fully recovered. And also from the bacterial count, it's also gave us the same result. So the summary for the antibiotics part, we have made these formula-defined dual ligand capped nanoclusters. As powerful antibacterial materials, we have discovered optimum ratio to afford the nanocluster with good potency and also biocompatibility. They can actually exhibit several different modes and then give good potency against gram-positive multi-drug resistant bacteria. Our cluster can significantly reduce the antibiotics required to treat MDR bacterial resistant infection. So it has the potential to revitalize the efficacy of the frontline biotics. And combine the gold nanocluster with antibiotics can be a cheaper, faster, and potentially longer lasting alternative to new antibiotics to address the bacterial antibiotic resistant challenges. Finally, I'd like to basically thank the people, especially Dr. Yuan Guo, Emma, Dashita, James, who did most of these glycan nanoparticle work, and this has been followed by other group members, which is currently undergoing. And glycan nanoparticle work, it's in collaboration with Yuan Guo, who is now academic, and Liz and Bruce Temple, who helped us on the sugar synthesis, and Nicole, who helped us on the EM studies, and Stefan from Germany, who helped us on the antiviral kind of evaluations. And the antibacterial work is mainly done by the student, PhD student Dejian, who is a joint PhD student between Leeds and Sustek in Xinyu Jiang's group. And of course, the financial support mainly from the BBSC, Wellcome, and Marie Curie's. Here's an advert. I do have 2 basically PhD positions open in my group to continue with this work. The first is antibiotic nanoclusters. These are funded by the Brack Center and also the glycan nanoparticles. And these are funded by the Zhou's BBSRC-DTP. If you are interested, then please get in touch. And thank you for listening. And I'm happy to answer any questions. Yeah, these are some of the photos from my former groups and our group parties, etc. Sorry for a little bit overrun of the time.
No worries at all, Professor Zhou. Thank you very much for a very, very engaging talk. If anyone has any questions, if they would just like to ask them in the chat or raise their hands, that would be great. If not, then I just have one quick question, is what would you say you think is the most rewarding part of this whole journey?
Sorry, can you repeat the question, please?
What do you think has been the most rewarding part of this whole sort of experiment and, you know, journey?
Yeah, hopefully, I think the antibiotics, nano antibiotics, that is, yeah, is really one of the rewarding kind of project, because we found, yeah, if you are really developing new antibiotics, then that will cost you billions for each of them in order to be able to bring into the market. And if we can really use these nano antibiotic to resensitize these basically antibiotics against the resistant bacteria, and effectively, yeah, you can save billions and make it much more treatable for these even resistant bacterial infections. And of course, the sugar nanoparticles, those are also actually quite important. One thing we can learn from this pandemic is if you're developing these vaccines or antibodies, the virus can evade quite quickly. But their sugar, it's not really changing that much. So, and also in this case, if we can use a glycoconjugate to block the virus to be able to reach to the host cells, then these bacteria will have low chance to to be able to develop resistance, etc. So that is also actually quite quite important, and also hopefully we might be able to use our nano-related research to really make some impact to the biomedical areas as well.
Yeah, definitely. I think it's, you know, very clearly really important work that you guys are doing. And yeah, really, really interesting. So if no one else has any questions, then say another big thank you for Professor Zhou for coming in and talking to us today.
Okay,
And,
my pleasure.
Yeah, hope you have a brilliant rest of your day. Thank you.
You too.
Thank you so much for the talk.